Hello everybody, here's your review for the first half of Unit 5 on the urinary system, and of course a review for the respiratory system will follow. And as always, I've got a series of some example questions here to help us talk about some of the topics and review some of the highlights. Just remember, there's really not a lot of point in trying to memorize the answers to these questions because they're not identical to the test questions. This is more about reviewing the concepts. It just helps us talk our way through some things here. All right, first question, what is the functional unit of the kidney? In other words, what part of the kidney is concerned with filtering the blood, creating that filtrate, and then concentrating it into urine? Where does all that take place? And hopefully you are thinking of the nephron. The nephron, now the kidney contains, nephrons are microscopic, so the kidneys contain many gazillions of nephrons, all of which are involved in filtering the blood and creating the urine, which will contain the wastes that need to be excreted from the body. All right, so just to review some of these structures, I've pulled in a few diagrams from your textbook. And all right, so this squiggly yellow structure that you see over here is an example of a single nephron. But notice over here, look really closely at the figures in your textbook, because here we've got a cross-section through the kidney, and it's showing us that we are blowing up this little tiny small window here to reveal what a single nephron looks like. You're going to have these nephrons all over the cortices of both of your kidneys. All right, and what do we have here in red? We hopefully recognize those as arteries when we're thinking about systemic circulation. All that stuff we learned about back in the cardiovascular unit still applies here. And this is an interlobular artery, so you have those also called cortical radiate arteries. So those extend, kind of flare outward all along the cortex of each kidney, so they are carrying oxygenated blood. Periodically, those are going to exit onto, and this is an afferent arteriole, so those bring blood to the capsule here where filtration will take place. That actually, the afferent arteriole leads to the squiggly red vessel that you see on the inside. That's called the glomerulus. And remember, the glomerulus contains blood. It's actually a blood vessel, a capillary. And the blood gets filtered in there, plasma gets squeezed out, and that's what we call the filtrate. That gets captured by this glomerular capsule that is surrounding it. That's also called Bowman's capsule. The filtered blood then leaves through the efferent arteriole that you see there. All right, so now you've got all this fluid that got squeezed out of the glomerulus there, and again, this is happening all over the cortex of each of your kidneys constantly throughout the day. That filtrate will pass first into a proximal convoluted tubule that you see here. That leads to the loop of Henle, also called the nephron loop, and you got a descending limb here, ascending limb over here. That leads to a distal convoluted tubule, which connects to a collecting duct. And so here's a single collecting duct. You can see all these stumps coming off of it because there are distal convoluted tubules of other nephrons connecting into that. The collecting duct leads to, eventually over here, your minor calyces and your a minor calyx, a major calyx, and down into the renal pelvis. And then the urine's going to flow from there into a ureter and on toward the bladder. And here's not all of your nephrons in the kidney. Kidneys look the same. Most of them, though, look like this. So over here, you're seeing a nephron in 2D, like it's been ironed and flattened out so that you can see all the different structures. In reality, they're all wadded up like this, and they kind of look like a jumble of spaghetti. But most of the nephrons in your kidneys primarily are most of them are located in the cortex. Those are called cortical nephrons. Again, they're all kind of wadded up like that. And then you have some that are called juxtamedullary nephrons, and those have the really long loops of Henle that go way down into the renal pyramids or the medulla. They hang way down in there. And remember, those are concerned with establishing that salt gradient where you have a lower sodium concentration here close to the cortex, and the deeper you go down into the medulla, the salt concentration gets higher and higher and higher. And that's important because, as we've learned, water follows sodium, so you have less sodium here, more sodium down in here, And as urine is flowing through the collecting duct, as it goes deeper and deeper and deeper into the medulla, that higher salt concentration draws more and more and more and more water out of the urine as it's passing through here. And that occurs as needed when you need to concentrate your urine and retain more water to help you maintain hydration of your tissues. All right, in purple here, those represent capillaries that surround the nephrons. So those are called peritubular capillaries when they surround a cortical nephron. The peritubular capillaries that surround the really long loop of Henle on a juxtamedullary nephron have a special name. They're called vasa recta. They kind of look like drapes. And they're also involved in helping to maintain that salt gradient that we were just talking about. If you look really closely at the diagram here, where do these peritubular capillaries come from? Here is an afferent arteriole bringing blood into the glomerulus inside, surrounded there by the Bowman's capsule. The filter blood leaves through an efferent capillary, and notice then that branches into these peritubular capillaries that surround the cortical nephron there. And then eventually those lead back over here to an interlobular vein. So the blood is now deoxygenated. It has given up oxygen and nutrients to all these kidney tissues. These are all active, metabolically functional tissues, so they need oxygen gas and nutrients just like other parts of your body. So just kind of putting some concepts together there that we've learned in the class. All right, so when we studied urine formation and we talked about reabsorption of substances out of the filtrate and back into the blood, back into your tissues, that's generally, it's the peritubular capillaries that are reabsorbing those things. So we also talked about tubular secretion, so things moving out of the blood and out of our tissues and into these different parts of the nephron over here. And so in a lot of cases, those substances are moving out of the peritubular capillaries, briefly into the surrounding interstitial fluids or tissue fluids, and then on from there into the nephron itself. So that's why you got to have these capillaries really tightly wrapped around the nephrons. All right, then really zooming in here. Now zooming in on Bowman's capsule and the arterioles and the glomerulus there, what's going on? Remember, your afferent arteriole is leading toward the glomerulus. It's been cut open here on the inside of our renal corpuscle here. And then the filter blood exits through the efferent arteriole. Efferent, E for exit. A for arrive. This is incoming blood, this is outgoing blood. And remember, here in your glomerulus, this is a blood vessel. You have blood in here, you don't have urine inside this blood vessel. The fluid gets squeezed out due to blood pressure. It gets squeezed out or filtered out through the walls of the glomerulus, and then it gets captured in here by the Bowman's capsule or the glomerular capsule. So in here in these spaces in that lumen you see there, that's where you have the earliest urine or the glomerular filtrate. And then we also learned a little bit about the juxtaglomerular apparatus, these cells over here that are highlighted in purple, and you've also got these granular cells located kind of here in brown. And these structures are concerned with maintaining glomerular filtration rate, and they're also involved in regulating blood pressure, which we'll review a little bit more about shortly. All right, next question. What structure is the first location where early urine or filtrate can be found? And hopefully you're thinking the glomerular capsule. Don't say the glomerulus. You have blood in the glomerulus, not urine. It's the fluid gets squeezed out of the glomerulus and into the surrounding capsule. That's where you have your first blood. How would you your first urine, sorry. How would you compare the volume of glomerular filtrate produced daily to the volume of urine actually excreted? Is the filtrate volume much higher, approximately the same? Is your urine volume much higher, or is your filtrate volume just a little bit higher? And hopefully you answered A on that one. All these nephrons in both of your kidneys are constantly filtering the blood, capturing lots of fluid, lots of plasma gets squeezed out into the Bowman's capsules, and that's your earliest filtrate. But then remember, from there, your nephrons and your collecting ducts are concerned with making sure you reabsorb the good stuff like amino acids and sugars and things that you need to feed your cells, reabsorbing electrolytes at the appropriate levels to help you maintain homeostasis of those electrolytes, reabsorbing water on an as-needed basis to help you maintain your fluid balance as well. So in general, the urine gets concentrated a lot as it's passing through the nephrons and the collecting ducts. So your filtrate volume is much, much higher than the volume of urine you actually excrete out of your body. And in fact, I think in our lectures we talked about how your blood is, by healthy kidneys, it's filtered 60 times a day. So that lets you know how important blood filtration is. All right. Next question. Which urine formation mechanism adds additional substances to filtrate after it leaves Bowman's capsule? So let's go back to our diagram over here. Let's look back over here at the squiggly one over here. Okay, so you've got your Bowman's capsule over here captures this fluid that gets squeezed out of the glomerulus, out of the blood, passing through the glomerulus, and that's going to contain blood, that's blood plasma, and smaller molecules, waste, so you're going to have glucose in there. I mean, any kind of smaller building block type molecule will be in there, various electrolytes and so forth. And a lot of those are good things you don't want to pee out. And so as that filtrate is passing through these structures, the good stuff gets reabsorbed back out of the nephron tubules and into the surrounding tissue fluids, and then ultimately into these peritubular capillaries, because that's going to bring them back, those substances back into the blood. That's reabsorption. Secretion is the opposite. So there are some substances where after the filtrate has left the capsule, and while it's passing through the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, some additional substances are added to the filtrate, and that's called tubular secretion. So if we return to our question over here, which urine formation mechanism adds additional substances to the filtrate after it leaves Bowman's capsule? And so hopefully you are thinking tubular secretion. Some of that terminology you have to really think about. Reabsorption, you are reabsorbing good stuff. Tubular secretion, you are getting rid of additional things that the body needs to excrete or remove. All right, you would not pick glomerular filtration here because that actually takes place in Bowman's capsule, and urinary excretion is not going to be the correct answer on that one either. All right, which of these would not be included in glomerular filtrate under normal circumstances? And I guess actually I need to, let me rephrase this, because this is brand new, hot off the press, and let me, no, I'm sorry, we're good. I'm thinking something else. Never mind, you can tell this is a brand new video I'm making. Which of these would not be included in the glomerular filtrate under normal circumstances? So think about the blood passing from the afferent arteriole into the glomerulus, and you've got that hydrostatic pressure, that blood pressure that's forcing the fluid and smaller molecules out. If these structures, these glomeruli, these little tiny capillaries are functioning properly, what would be excluded from the filtrate? What would not leave the blood and become filtrate? Do we think it's plasma proteins, electrolytes, water, wastes such as urea? And think about big molecules can't get squeezed out of the glomeruli. They're too big. Smaller substances, though, do get filtered out. So our answer there, plasma proteins. Remember, those are large organic molecules, and they help maintain what's called the colloid pressure, and they contribute to the osmotic pressure that draws water back into the blood and helps us maintain our blood volume. So proteins in healthy kidneys are too big to get filtered out. Now, people who have damaged kidneys, like kidneys that have been damaged due to years of high blood pressure, so the walls of their glomeruli are leaky. They may actually have protein that gets into the urine, so that can be a sign of kidney damage. Electrolytes are small, they're generally single charged atoms, so those are small on the atomic and molecule type scale, so they're going to get filtered out. Water, of course, gets filtered out. And waste such as urea, we want to get rid of waste, so those are going to be filtered into the urine as well. All right, higher blood pressure will cause, this gets into the glomerular filtration rate, what volume of fluid per hour is being filtered out of the blood. That's your glomerular filtration rate. If your blood pressure is higher, so if we go back and look at our diagram over here, blood's coming in through the afferent arteriole, comes into the glomerulus. So the hydrostatic pressure, that blood pressure, is the main force that's squeezing fluid out of the glomerulus and into the surrounding Bowman's capsule. So if your blood pressure is higher, it's going to cause more fluid to be squeezed out. So higher blood pressure will actually increase your glomerular filtration rate. All right, now don't try to overthink or overcomplicate with a question like that, because another thing that we studied in this chapter and something we studied back in the cardiovascular part of the course was, okay, when your blood pressure goes up, your body is going to respond to try to maintain homeostasis. Okay, so, but when you see a question like this, just take it at face value. Don't think too many steps ahead. Just go with what it says here. Blood pressure has gone up. What is that going to do to your glomerular filtration rate at that moment? It's going to increase it. Don't think ahead to, okay, well, but if the blood pressure is higher, then the kidneys are going to respond because they've got that intrinsic mechanism that's going to lower the glomerular filtration rate. Yes, that's true, but initially, what is that higher blood pressure going to do? It's going to cause a higher glomerular filtration rate. Hopefully that higher blood pressure and that higher glomerular filtration rate will get corrected to help you maintain homeostasis. Which of the following would not be reabsorbed under normal circumstances? So something you just need to keep in mind, again, I know the terminology can be a little bit difficult. We reabsorb the good stuff, we secrete and excrete the bad stuff when it comes to urine formation. Or secretion can also be used to control pH as well, as we'll talk about a little bit later. All right, but which of the following would you not want to reabsorb? Glucose, hopefully we know that's good stuff. Urea is a waste, so we want to get rid of that. Amino acids are good. We need amino acids to build our proteins and our cells. Water, we reabsorb most of the water back out of the filtrate. It goes back into our body's tissues before it is excreted as urine. So your answer there is going to be B. Urea is a waste that's generated mostly from breaking down proteins, and we need to pee that out. That's easy to remember because urea sounds like urine. Same thing with uric acid. Uric acid is a waste product from breaking down nucleic acids, and it also sounds like urine, so it sounds like a waste. That's something you want to pee out of the body. All right, hormone question. Which hormone directly triggers more water to leave the collecting ducts and enter the surrounding tissues and capillaries? All right, and hopefully you're thinking ADH, anti-diuretic hormone. That's what it does. It acts its target. And we learned about that hormone back at the beginning of the semester when we were studying endocrine, but now we know more details about the kidneys, and so we've learned more specifically, well, where is it acting? It's acting on those collecting ducts. It triggers the collecting ducts to produce aquaporins, which are water channels, and the cells there that line those collecting ducts stick those water channels in their cell membranes, and that allows water to flow out from the collecting ducts and into the surrounding tissues, and then they get taken up by that excess water, gets taken up by the capillaries in there as well, so it winds up back in the bloodstream. So that's the job of ADH. ADH triggers the collecting ducts to reabsorb water back into the surrounding tissues and eventually into the bloodstream. And of course, we also talked about renin and aldosterone as well. We'll review those shortly. And ANP, atrial natriuretic peptide, remember that one? Natriuretic refers to sodium. Uretic, you're going to excrete sodium. Remember, water follows sodium, so more sodium in the urine. It's going to draw water into the urine from surrounding tissues, and so that would actually increase your urine volume. Remember, we use ANP to help reduce fluid volumes in our body, in our bloodstream, to help lower blood pressure when needed. So ANP actually does the opposite of ADH. All right, here's a question that's going to tie in pH to what we've been talking about. If tubular secretion of hydrogen ions increases, what will happen to a patient's body fluid pH? All right, so to answer this question, we have to understand the relationship between hydrogen ion concentration, and hopefully my little pen over here is going to work. Remember, if we put hydrogen H+, we put brackets around it, that's referring to the concentration of those hydrogen ions. If the concentration of hydrogen ions increases, what happens to the pH value? It actually goes down. There is a mathematical reason for that. You'll learn it when you take a chemistry class if you have to take one. On the other hand, if your hydrogen ion concentration drops, your pH value goes up. And we associate low pH with acidity. We associate higher pH with alkalinity. That's just some terminology, some things that you need to learn. Okay, so let's think about what's going on in this patient. Tubular secretion of hydrogen ions increases. So now we have to think about what's happening to the patient's body fluid. Tubular secretion means more hydrogen ions are being sent into the urine so that you pee them out, you excrete them out of the body. So what's going to happen to the hydrogen ion concentration in this patient, in the patient's body fluids? If you're peeing out more hydrogen ions, the concentration of those hydrogen ions in the patient's body fluid will go down. So what's that going to do to the pH? Low hydrogen ion concentration means we have an increase in pH. All right, so hopefully there you would turn off my pen here. Oops. Select A, your pH will increase. So that might happen if you were experiencing a case of low body fluid pH and you need to try to correct it. So if you have low body fluid pH, you've got too many hydrogen ions around, maybe you need to excrete more of those out of the body to help bring your pH back up. Okay, so we studied glomerular filtration rate. That's very tightly regulated in the kidneys to try to make sure you are filtering the fluid in the blood at a fairly consistent rate. And of course, all that's going to be controlled based on your blood pressure and your body fluid balance needs and so forth. Very critical part of homeostasis to keep us alive. But how fast glomerular filtration occurs, how much fluid per hour is actually being squeezed out of the blood, is a very important thing that has to be controlled. And we have what are called intrinsic controls. So that's just a function within the kidney itself that's essentially automatic that helps control GFR under normal circumstances. Then you have longer term control, and that's where we get into how the nervous system and the endocrine system help control the GFR and also your blood pressure on more of a long-term basis. All right, if your blood pressure is going up, we've already talked about how that is going to increase your GFR because you're going to have higher blood pressure pushing against the walls of those glomeruli in your kidneys, and that's going to force more fluids through the walls and into the surrounding capsule. All right, but you have smooth muscle cells in the walls of your afferent arterioles, those incoming blood vessels, and they're able to sense, okay, blood pressure is high, so the walls of the arterioles, that fluid is pushing against them, and the smooth muscle cells in there are going to react. So if your GFR is too high, how are you going to lower it? You've got to reduce the amount of blood that's actually making it to the glomerulus. So to do that, then the smooth muscle cells in the walls of your afferent arterioles contract or constrict. It makes the diameter of the afferent arteriole smaller, and that allows less blood to go into the glomerulus. Okay, and then that's going to lower your glomerular filtration rate. Okay, go back and look at your diagram and so forth and make sure that makes sense. So this is part of the intrinsic control of GFR. These smooth muscle cells in the walls of the afferent arterioles will contract or constrict if the GFR is too high, and that's going to help reduce it by permitting less blood to enter the glomerulus. Less blood in the glomerulus lowers the hydrostatic pressure in there, less fluid gets filtered out. So we want to restrict blood flow. Now on the other hand, if your GFR is too low, you don't have enough blood in the glomeruli in your kidneys, then those smooth muscle cells will relax. They'll open up the diameter of the afferent arterioles, which will let more blood go into the glomeruli. So then more fluid will get filtered out of the glomeruli and into the surrounding capsules. All right, low mean arterial pressure, so if you have low blood pressure, will lead to an increase in all of these except what? So this gets back into longer term control. The kidneys are very concerned with glomerular filtration rate. Blood pressure plays a big role in that, so your kidneys wind up being one of the main organs involved in long-term blood pressure control. So one of these is not associated with, or one of these you would not expect to be at higher levels if you had low blood pressure. And hopefully you're thinking C, the ANP, the atrial natriuretic peptide. We just talked about that. All right, when blood pressure is low, your glomerular filtration rate will be low because you don't have as much blood flowing as quickly into the glomeruli where filtration will occur. That causes renin to be released. You've got those granular cells we just took a look at that are right there close to where the afferent arteriole meets the glomerulus. Renin is an enzyme that converts, go back and look at your pathways. You always have angiotensinogen floating around in your blood. Renin clips that and creates angiotensin 1. And then remember, you have an enzyme called angiotensin converting enzyme that converts this to angiotensin 2. Angiotensin 2 does all kinds of things to help increase your blood pressure, so be sure you go back and review those. One of those is it triggers aldosterone release from where? The adrenal glands, the cortex of the adrenal glands. And aldosterone, remember that story, triggers an increase in sodium reabsorption out of the filtrate and back into your tissues in your bloodstream. Water follows the sodium, which increases your blood pressure. Okay, so you may need to go back and listen to that again, but that story needs to make sense to you for you to have a good understanding of how blood pressure and this fluid regulation, this fluid balance and so forth that's so tightly linked to blood pressure actually works. All right, a pretty common type of drug, a lot of patients take these, an ACE inhibitor drug will cause, select all that applies. All right, A stands for angiotensin converting enzyme. So just to review again, if your blood pressure is low, renin gets secreted by the kidneys. And renin is an enzyme, and it converts angiotensinogen into angiotensin 1, and I'm just going to write that as A1 up here. Angiotensin 1 gets converted into angiotensin 2 by this enzyme called ACE, which stands for angiotensin converting enzyme. So remember, enzymes help catalyze or carry out chemical reactions. So this enzyme ACE, it's kind of like scissors. You have angiotensin 1, you cut the end off of it, it becomes angiotensin 2, and now it has all kinds of additional functions. Angiotensin 2 increases blood pressure in a number of different ways. All right, so let's think about this. If you're taking a drug that inhibits ACE, you're going to have more or less angiotensin 2 around. You're going to have less. If you don't have the enzyme around that makes it, you're going to have less of this. So your angiotensin 2 levels will drop. And so we just said angiotensin 2 does lots of things, which you need to go back and review to help raise your blood pressure. So let's take a look at these. Would we expect a decrease in ADH? Actually, we would. If you go back and look, angiotensin 2, one of the things that it does is it increases the ADH release from the posterior pituitary gland. And so if you have less angiotensin 2 around, you're going to have lower levels of ADH. Remember, a person would take an ACE inhibitor drug because they have high blood pressure or low blood pressure. Hopefully you're thinking high blood pressure. If you take an ACE inhibitor, it's because you have higher blood pressure, and then they're trying to help keep your this renin, angiotensin, aldosterone system under control so that it doesn't try to raise your blood pressure even more than it normally would be. Would you have a decrease in aldosterone? Yep. Hopefully you're thinking yes on that, because angiotensin 2 tells the adrenal cortex to secrete more aldosterone. So if you have less of that, you're going to have less aldosterone. Constriction of the walls of the arterioles. We'll say no on that. Angiotensin 2 is one of the things that triggers the walls of your arterioles to constrict. Remember, that helps increase peripheral resistance, which helps increase your blood pressure, and we don't want that to happen. Would you have a decrease in angiotensin 1? I would say no. ACE takes that, angiotensin 1, and converts it to angiotensin 2. So if you don't have ACE working properly, I would assume your angiotensin 1 levels would probably go up because it's going to accumulate in all likelihood. An increase in angiotensin 2? No. And how about higher blood pressure? Hopefully not. If you're on an ACE inhibitor drug, the whole point is to try to lower your blood pressure. It doesn't work in everybody because blood pressure regulation is very complex and lots of things impact blood pressure. Your answers there are A and B. All right, another urine formation question. The countercurrent mechanism involving the juxtaglomerular nephrons and the vasa recta. So again, those are the nephrons where the loop hangs way down into the medulla. And so earlier we talked a little bit about what the function of those nephrons were. And if read through the answers and try to make your choice. And hopefully you picked D. The job there is to maintain a strong salt gradient in the renal medulla, where you have a lower salt concentration closer to the cortex. The deeper you go down in the medulla towards the renal pelvis, the saltier the gradient gets, and that helps suck more and more and more and more water out of your collecting ducts. It helps concentrate the urine when you need to. That's under the influence of ADH. So if you don't need to retain more water in your body. Like you've been chugging water all day and you haven't been sweating, exerting yourself in any way. As you guys know, your urine volume goes up and your urine looks more dilute. That's because ADH is not being secreted to tell your collecting ducts to reabsorb more water. Okay, what effect would an ADH inhibitor such as alcohol have on your urine volume? All right, so if you inhibit, remember ADH does what? It concentrates the urine. It triggers water to be reabsorbed out of the urine. So if you if you're consuming a lot of alcohol, ethanol alcohol like you have in beer and wine, liquor, inhibits ADH, which means you're not going to be able to concentrate your urine. So hopefully you're thinking increases urine volume. And I know none of you guys have ever consumed alcohol, but if you ever do, you might notice that you have an increased output of urine. Urine pathway, be sure you review the pathway of the filtrate through all of the structures we've discussed, all the way from the Bowman's capsule, all the way to the point where it exits the body. So what structure or structures occur after the external urethral sphincter in the path of urine flow? Select all that apply. So let's go through these. Go back and look at the diagrams and the pictures, because you might get fooled by at least one of these on here. How about the trigone? Some people say trigony. If you go back and look at the diagrams, that's the area within the bladder. It's formed by the two openings where the right and the left ureters open up into the bladder. And then here is your internal urethral sphincter, where the urine is going to flow out of the bladder and into the urethra, extending there away from the bladder. So that little triangle there, it's called trigone, or some people say trigony, because those three openings kind of form a triangle. All right, so no, that does not happen after the external urethral sphincter. How about urethra? Now you might be tempted to say no, we're at the external urethral sphincter, so no, the urine's going to leave the body after that. Nope, go back and look at the pictures. The external urethral sphincter is located in the wall of the muscular wall of the pelvis, basically. And so you have additional urethra after that structure. So in a male, you've got the entire part of the urethra that passes through the penis to go through. In a female, you've got the little short stretch of the urethra that extends from the muscular wall of the pelvis to that external urethral orifice. So you do have more urethra in both males and females. Of course, you've got the external urethral orifice. That's the opening to the outside of the body. Ureter, no, you've already been through the ureters. Internal urethral sphincter, nope, that's going to be up here. There's your internal urethral sphincter. External down in here. Muscular wall of the pelvis there. And then again, you've got more urethra. Little short stretch for females, longer in males, because that's the part of the urethra that's going to pass through the penis. All right, so our answers there were B and C. Okay, a little bit on acid-base balance. And again, this is a topic that sometimes gives some students some trouble at first. You just have to practice with it. And don't lose your information from this class on this topic, especially if you're going to nursing school, if you're going to go to medical school, because it will, you want to hang on to the information from this class because it'll help you down the road. Every semester we want to send in acid-base balance information to nursing students who are freaking over about it when they get into nursing school and have a whole lot more to deal with in their classes at that time. All right, a key thing to think about with acid-base balance is don't forget your chemical equation. CO2 in body fluids rapidly and just naturally combines with water to form H2CO3, and then that quickly breaks apart into a hydrogen ion and this molecule over here, bicarbonate. So that's carbonic acid, and that's bicarbonate. I'm just going to write bicarb. Okay, so we have to always keep this equation in mind. But what I usually tell students to focus in on, the more CO2 that your body is producing, this is a back and forth chemical equation. It's kind of like a seesaw that goes back and forth. The more CO2 that your body is producing, it tends to drive the equation in this direction in a greater amount. The more you go that way, the more CO2 around, the more carbonic acid you have around. Acids are compounds that release hydrogen ions. And remember what we just said, if you increase, you have higher hydrogen ion concentration in your body fluids, pH goes down, and that's acidic. That's associated with acidity. The reverse, lower hydrogen ion concentrations, that raises your pH. And that's associated with alkalinity. Compounds that do that are also called bases. All right, if you kind of look at your equation here, acids like carbonic acid, the reason they're called acids is because they dissociate or break apart, and they release hydrogen ions into our body fluids. So the more of this you have around, the more hydrogen ions you're going to have in your body fluids. That's going to lower your pH. Your body fluids become more acidic. On the other hand, bases like bicarbonate soak up hydrogen ions. So instead of those hydrogen ions freely floating around in your body fluids, they get soaked up and they send the reaction back in this direction. And if they're soaking up your hydrogen ions, that is going to raise your pH. Your body fluids are going to tend to become more alkaline. And so I know some of this is kind of difficult to grasp at first. You've got this seesaw, this what's called an equilibrium going back and forth. But the easiest thing to remember is anything that increases the CO2 is going to drive the equation this way. So you're going to have more hydrogen ions around and a lower pH. Anything that causes an increase in bicarbonate ions will drive the equation back this way, because those excess bicarbonate ions are going to soak these guys up, and that's going to lower the hydrogen ion concentration in your body fluids and raise your pH value. And honestly, if you just kind of think about it on those levels, you can save the more complex thoughts about pH balance and chemical equilibrium and so forth for your chemistry classes down the road if you have to take them. Try not to overcomplicate. So let's go back and look at this question. If less secretion of bicarbonate ions into urine occurs, what's going to happen to your body fluid pH? Remember, we said secretion means you're getting rid of the bicarbonate ions. All right, so we're secreting less of those into the urine, which means we are hanging on to more of those in our body fluids. And what did we just say bicarbonate ions do to our pH? Bicarbonate ions love to soak up. They bind with hydrogen ions. So your hydrogen ion concentration is going to go down, which means your pH will go up. Your pH will increase. Just, if you have to, just memorize that. Bicarbonate ions in greater numbers increase your pH. CO2 leads to a decrease in pH. That's the easiest way to learn it. If a patient has metabolic alkalosis, so we learned about respiratory acidosis and alkalosis, and then metabolic is for other reasons. When you develop alkalosis and acidosis due to other reasons besides your breathing rate and depth. So metabolic alkalosis, so what's happened to the pH? It's gone up. How might this be compensated? Remember, compensation generally refers to, all right, if you have metabolic problem, can you compensate it with the respiratory system. So can you compensate by adjusting your breathing rate and depth? And we talked about that a little bit during the respiratory part of the course. Or if you have respiratory acidosis or alkalosis, it's usually the kidneys that make the adjustment. Can you adjust your bicarbonate ion levels to try to correct the problem? So compensation refers to trying to correct the problem with the opposite body system. Okay, so if we've got a patient who has a increased pH value in their body fluids, what do we need to bring the pH back down? So if we're thinking about how the respiratory system might accomplish this, what do we need to bring pH back down? We need more CO2 in the body fluids. Does that make sense? Because if you retain more CO2 in your body fluids, that's going to, CO2 functions as an acid. It gets converted to carbonic acid by reacting with water. So that is going to help lower the pH. All right, so then we're thinking about the respiratory system. How does the respiratory system make an adjustment so that we retain more CO2 in the body? We don't breathe it out. We need to, hopefully you're thinking, decrease respiration. So you're going to lower your breathing rate and depth. That will leave more CO2 in your body fluids, in your bloodstream, which hopefully will help lower pH. All right, I'm going to do a couple of acid-base balance problems just to kind of walk us through, and then be sure you go back. If you're still not understanding this, use the handout that's on Blackboard, which has lots of example problems in it, and go back and review the video lecture where I went over some example problems as well. Just practice until you get it. There are 2 of these on the exam, so you do need to know how to do these. All right, diagnose this patient based on these clinical test results. All right, so, and I will give you the normal values. These are the normal values for carbon dioxide levels, which are given in millimeters of mercury. That's what Hg stands for. That's a partial pressure like we learned about when we covered the respiratory system. And bicarbonate ions there, those values are given in something called milliequivalents per liter. That's just the units referring to the concentration of those ions in our body fluids, and don't worry about what a milliequivalent is. Just know those are the units for measuring bicarbonate ions. All right, so what we have to do, first of all, pH is 7.51. Now you should know the normal pH range for human body fluids by now. It's 7.35 to 7.45. So first we have to figure out, well, does this patient have a problem? And 7.51, the pH is too high. So is that acidosis or alkalosis? Hopefully you're thinking alkalosis. And then we have to go back and look at the CO2 levels and the bicarbonate levels and see which of those matches the problem. All right, so the CO2 level 32, this patient has low CO2, and how about 24 for the bicarb? That's within the normal range. All right, so does low CO2 match alkalosis? And hopefully you're thinking yes. High CO2 would match acidosis because the pH would be lower. CO2 functions as an acid. If you have low CO2, you don't have enough acid being produced in your body fluids, and that's going to cause your pH to go up. So which system are we blaming here? We're blaming the respiratory system. So this is going to be respiratory alkalosis, because the respiratory system seems to be the problem. Think about it more deeply. What's going on here? You're probably, CO2 levels are too low. You might be hyperventilating. You might be breathing too quickly, releasing too much CO2 out of the body and into the air. The next thing we have to know, is it compensated, partially compensated, or not compensated? So look at the other value. In this case, it's within the normal range. So bicarbonate ions, the concentration of those is adjusted by the kidneys. through secretion. And so at this point it appears as though the kidneys aren't doing anything because our bicarb levels are within the normal range. So we'll say uncompensated in this particular case. Okay, let's take a look at this last problem here. We've got a patient with a pH of 7.28, CO2 at 48, bicarb at 29. So what's going on here with this one? pH of 7.28, is that acidosis, normal, or alkalosis? Hopefully you're thinking acidosis because the pH is too low. All right, CO2 is at 48. Is that too high or too low? We got high CO2. And go ahead and think about what that would do to the pH of your body fluids. All right, high CO2 is going to raise your pH or lower it? It's going to lower your pH. CO2 functions as an acid in your body fluids. How about the bicarb? Too low or too high? It's high. What will high bicarb do to your pH? It will increase it. Okay? All right, which of those two abnormal values there is associated with acidosis? Hopefully you're thinking CO2, the abnormal CO2. So that tells us that this patient has respiratory acidosis. CO2 levels are too high, so probably the patient is not breathing at a fast enough rate or not deep enough. And okay, the bicarb is too high, so the bicarb levels are trying to bring the pH back up. It's trying to increase it. We are not yet back into the normal range, so is that going to be partial compensation or full compensation? And hopefully you're thinking partial compensation. And again, bicarbonate levels are adjusted by the kidneys through secretion. So the kidneys here are secreting less bicarbonate than they normally would into the urine. We're trying to hang on to bicarbonate ions and keep those values up in an effort to try to bring the pH back up into the normal homeostatic range. But that has not yet worked well enough for this patient's pH values to return to the normal level. So we say it's partially compensated, trying to correct the problem but has not achieved it yet. When would you have a case of full compensation? Let's say full compensation means you're back in the normal range. So if that value was 735, that would be a case of full compensation. So you'd still, by looking at the values, you'd still be able to figure out what went wrong because you're closer to the acidic side of things, so that would tell you had a respiratory acidosis problem. But since the high bicarb levels had brought you back to 7.35, you're back to the normal homeostatic range. At that point, you can refer to it as full compensation. If you have a full compensation problem, the pH value will be 735 or 745 on the other side for compensated alkalosis. We're not going to get into a real complex clinical problem where you have compensation going on, but your pH value is like right in the middle, and you can't really easily figure out what the problem is with the patient. You can save those for medical school if you go to medical school. Okay. All right, I think that's it for the review. Good luck with the urinary system, and let me know if you have any questions. I will be posting a video of the respiratory system next to finish out Unit 5 and the course. Thank you.